This is Chapter 21, Module 1, Intro to Blood Vessels. The learning objectives of this module are 1. Distinguish among the different types of blood vessels and compare the differences in their structure and function, and 2. Explain the mechanisms that regulate movement of fluids between the capillaries and the interstitial spaces. Blood vessels are classified by size and histological organization. Arteries are vessels that carry blood away from the heart. Arterioles are the smallest branches of arteries. Capillaries are the smallest blood vessels and are the only place where exchange occurs between the blood and interstitial fluid. Venules are small veins that collect blood from the capillaries, and veins return blood to the heart. The largest blood vessels are attached to the heart. The pulmonary trunk divides into the right and left pulmonary arteries that carry blood from the right ventricle to the pulmonary circulation. The aorta carries blood from the left ventricle to the circulation. The smallest blood vessels are the capillaries. The capillaries have small diameters and thin walls. Chemicals and gases diffuse across the walls of the capillaries. The vessel walls have 3 layers: the tunica intima, the tunica media, and the tunica externa. The tunica intima has an endothelial lining and a connective tissue layer. In addition, in arteries, an internal elastic membrane lines the outer margins of the tunica intima. The tunica media or middle layer contains concentric sheets of smooth muscle in loose connective tissue. The tunica media binds to inner and outer layers of the vessels. An external elastic membrane separates the tunica media from the tunica externa. The tunica externa or outer layer anchors the vessel to adjacent tissues in arteries. The tunica externa contains collagen and elastic fibers. In veins, this layer also contains smooth muscle cells. There are also small arteries and veins that supply the cells of the tunica media and tunica externa, called the vasa vasorum, otherwise known as the vessels of vessels. While arteries and veins run side by side and the 3 layers are present in both arteries and veins, there are some significant differences between the 2 types of blood vessels. Arteries have thicker walls and have a higher blood pressure than veins. A collapsed artery has a small round lumen or internal space, while the vein has a large flat lumen. The lining of the vein contracts, whereas the arterial lining does not. The artery lining has folds. The arteries are more elastic, and the veins have valves. We will talk first about the arteries. Arteries have an elasticity that allows them to absorb pressure waves that come with each heartbeat. Arteries can change diameter. The change in diameter is controlled by the sympathetic division of the autonomic nervous system. Vasoconstriction is caused by the contraction of arterial smooth muscles of the autonomic nervous system and causes the lumen to constrict, whereas vasodilation is caused by the relaxation of the arterial smooth muscles and causes the lumen to enlarge. The large arteries on the base of the heart are called the elastic arteries. The elastic arteries lead to medium-sized arteries called muscular arteries that lead to smaller arteries called arterioles. The elastic arteries are also called conducting arteries. They include the pulmonary trunk and the aorta. The tunica media of these elastic arteries have many plastic fibers and few muscle cells. The muscular arteries are also called distribution arteries. They are medium-sized arteries whose tunica media have many muscle cells. The arterioles are small and have little or no tunica externa. They also have thin or incomplete tunica media. The diameters of the small muscular arteries and arterioles change with the sympathetic or endocrine stimulation. Constriction of the arteries oppose blood flow. The arterioles are considered the resistance vessels due to the large number of arterioles that there are and their ability to constrict. As the resistant vessels, arterioles have the largest impact on blood pressure. Capillaries are the smallest vessels with the thinnest walls. Microscopic capillary networks permeate all active tissues. Capillaries are the location of all exchange functions of the cardiovascular system. Materials diffuse between the blood of the capillaries and the interstitial fluid. The capillaries structurally are just microscopic tubes of endothelial cells inside a basement membrane. They have no tunica media and no tunica externa. The diameter of the capillary is similar to that of a red blood cell. There are different types of capillaries, including continuous capillaries, fenestrated capillaries, and sinusoidal capillaries. Continuous capillaries have complete endothelial linings. Continuous capillaries are found in all tissues except epithelia and cartilage. Continuous capillaries permit diffusion of water, small solutes, and lipid-soluble material across the capillary walls, and they also block blood cells and plasma proteins from exiting them. Specialized continuous capillaries are found in the central nervous system and the thymus. These capillaries have very restricted permeability. In the brain, this restricted permeability helps to form the blood-brain barrier. Fenestrated capillaries have pores in the endothelial lining. They permit rapid exchange of water and larger solutes between the plasma and the interstitial fluid. They are found in the choroid plexus, endocrine organs, kidneys, and intestinal tract. Sinusoidal capillaries have gaps between the adjacent endothelial cells. They are found in the liver, spleen, bone marrow, and endocrine organs. They permit a free exchange of water, and large plasma proteins can escape from the blood and enter the interstitial fluid. Phagocytic cells monitor the blood for pathogens at the sinusoids. Capillaries are arranged in capillary beds. The capillary beds connect one arteriole to one venule. Venules are very small veins. The venules merge to form medium-sized veins that merge to form larger veins that bring blood back to the heart. The veins collect blood from capillaries in tissues and organs. Their diameter is larger than arteries, and they have thinner walls than arteries. Veins also have lower blood pressure than arteries. The large veins have all three of the tunica layers. They have a thick tunica externa and a thin tunica media. Large veins contain venous valves that are folds of tunica intima. These valves prevent the backward flow of blood. Compression of the veins by skeletal muscles push blood toward the heart. 30 to 35% of the blood volume is held in the heart, arteries, and capillaries. 60 to 65% of the blood volume is held in the venous system, with 1/3 of the venous blood being held in the large venous networks of the liver, bone marrow, and skin. Next, we will discuss the flow of blood in the capillaries and the factors that affect it. There are 3 important processes that move material across typical capillary walls. These 3 processes are diffusion, filtration, and reabsorption. Diffusion is the movement of ions or molecules from an area where their concentration is higher to an area where their concentration is lower. The difference between the high and low concentrations represents a concentration gradient. Diffusion occurs most rapidly when the concentration gradient is large. It also occurs most rapidly when the ions or molecules are small or when the distance for them to travel is short. Different substances diffuse across capillary walls by different routes. Water, ions, and small molecules such as glucose and amino acids can usually enter or leave the bloodstream by diffusing between the endothelial cells of the capillaries, or they can diffuse through the pores of the fenestrated capillaries. Ions such as potassium, sodium, calcium, and chloride diffuse across the endothelial cells through channels in the plasma membranes. Large water-soluble compounds are unable to enter or leave the bloodstream except at fenestrated capillaries. Fenestrated capillaries are at the hypothalamus, the kidneys, other endocrine organs, and the intestinal tract, and are the only place for these large water-soluble compounds to enter or leave the bloodstream. Lipids, like fatty acids and steroids, as well as lipid-soluble materials such as oxygen and carbon dioxide, cross the capillary walls by diffusing through the endothelial plasma membranes. Plasma proteins are normally unable to cross the endothelial lining anywhere, except in places like the liver, where there are sinusoids. Solutes can be pushed out into the interstitial fluid between the cells of the capillary wall in a process called filtration. Filtration is the removal of solutes from a solution as the solution flows across a porous membrane. The driving force for filtration is hydrostatic pressure. Hydrostatic pressure is caused by the blood flow as the blood pressure pushes the blood up against the walls of the capillary. Water and small solutes are forced against the capillary walls and pushed out of the capillaries into the interstitial fluid, leaving larger solutes and suspended proteins in the bloodstream. The solute molecules that leave the bloodstream are small enough to pass between adjacent endothelial cells of the capillaries or between the pores in the fenestrated capillaries. Filtration takes place at the arterial end of the capillary where the pressure is greater than at the venous end of the capillary. Reabsorption is another process that moves materials across the capillary walls. Reabsorption occurs as a result of osmosis. Osmosis is the diffusion of water across a selectively permeable membrane where water moves from high water concentration to low water concentration, or water moves from low solute concentration to high solute concentration. In other words, water diffuses towards the solution containing the higher solute concentration. There is a higher solute concentration inside the capillaries due to the plasma proteins that are more abundant in the blood than they are in the interstitial fluid. The osmotic pressure of a solution is an indication of the force of osmotic water movement. The higher the solute concentration of a solution, the greater is the solution's osmotic pressure. Therefore, the more suspended proteins there are in the plasma that can't cross the blood vessel walls, the greater is the plasma's osmotic pressure. In other words, the plasma proteins act as solutes that will pull water from the interstitial fluids into the blood vessels. The osmotic pressure caused from the plasma proteins is often called blood colloid osmotic pressure, or BCOP. Clinically, another term used for this is oncotic pressure. The movement of the water due to the osmotic pressure will continue until the solute concentrations are equalized between the inside and the outside of the blood vessels. As an overview, water moves continuously out of the capillaries and into the tissues. From the tissues, it will move into the lymphatic vessels and back to the heart, where it will be deposited back into the blood. This movement of water helps to ensure that the plasma and the interstitial fluid are in constant communication and mutual exchange, and it accelerates the distribution of nutrients, hormones, and dissolved gases throughout the tissues. As the blood flows from the arterial end of the capillary to the venous end, the blood pressure drops, and so less filtration and reabsorption will occur at the venous end of the capillary. It's important to understand how the net filtration and the net reabsorption are calculated. The net hydrostatic pressure is the difference between the hydrostatic pressure inside the capillary wall and the hydrostatic pressure outside the capillary. The blood pressure pushes the blood against the walls of the capillaries, but this is somewhat opposed by the pressure of the interstitial fluid against the wall of the capillaries. There's more of a push to get water out of the capillary than there is a push to get fluid into the capillary. The net hydrostatic pressure is the difference between those two forces. The plasma proteins in the capillaries create the colloid osmotic pressure that pulls water into the blood vessel, but there is also interstitial fluid colloid pressure from a small amount of plasma proteins in the interstitial fluid. More of a pull comes from the plasma proteins inside the capillaries than from the proteins in the interstitial fluid. The net colloid pressure is the difference between the pull from the interstitial proteins and the pull from the blood plasma proteins. There is more of a net push to move fluid out of the capillary than there is a net pull to move fluid into the capillary. The difference between the net push and the net pull is called the net filtration pressure. The net filtration pressure, then, is the difference between the net hydrostatic pressure and the net osmotic pressure. At the arterial end, the net filtration pressure will be much higher than at the venous end.
This ends Chapter 21, Module 1, Intro to Blood Vessels.